Toxins are perhaps the most obvious of virulence factors, and there's a wide variety of actions depending on what the target receptors, cells, or tissues are. We can broadly break toxins down into endotoxins, or lipopolysaccharide, LPS, which is a component of the gram-negative cell wall, and exotoxins, which are excreted effector molecules and are incredibly numerous. Examples of exotoxins include things like botulinum toxin produced by Clostridium botulinum, Shiga toxin produced by E. coli 0157H7, those enterohemorrhagic E. coli's, and cholera toxin. Some of you may be familiar with botulism toxin. It's actually used therapeutically as well as Botox. And Shiga toxin, which is a major concern for public health as it's associated with foodborne disease outbreaks. Superantigens are pathogen-associated structures which are able to non-specifically bind and activate the T cell receptor, resulting in a cytokine storm. T cells play an important role in recognizing the presence of potential infections in the body and are able to stimulate an inflammatory response when they notice something going on. If we consider the entire population of T cells in the body, the vast majority of antigens are only able to stimulate and activate a tiny proportion of them, somewhere between 1 in 100,000 and 1 in a million. So it's a very small number of these cells that actually respond in an infectious process. And the way that they respond is by secreting cytokines, things like IL-1, IL-2, interferon gamma, and tumor necrosis factor alpha, which leads to an inflammatory response. Superantigens have a unique ability to activate a much greater proportion of T cells. So instead of this very tiny percentage, it's actually maybe somewhere around 20%. And as a consequence, we get a massive release of cytokines, which leads to a massive systemic inflammatory response and ultimately shock and death. And finally, we have the production of enzymes. Bacteria can produce enzymes to degrade host tissues, to degrade antibiotics, to degrade metabolic products in their environment, oftentimes for nutritional acquisition, for nutrient acquisition, or for invasion of tissues. In this picture here, what you can see is actually a biochemical test, the hyaluronidase test. We have a streak of Staph aureus here growing next to a streak of Pasteurella maltosida. And what I hope you can appreciate is that the Staph aureus is actually breaking down the capsule surrounding the Pasteurella maltosida, leading to a much flatter colony appearance in this region. Now, in the host, this hyaluronidase enzyme is used to break down host tissues and facilitates invasion. Another great example is the presence of ureases. So urea is the nitrogenous waste of mammals, but bacterial ureases produced by uropathogens, so those bacteria that cause urinary tract infections, are able to break down urea into ammonia, which is, of course, the building block of amino acids and a valuable source of nitrogen in the urinary environment. We can also use the urease test as a biochemical test. So on the left here, we have a negative test. You can see the tube has this yellow color. And in the center, we have a urea-positive organism that's bright pink. After these descriptions, you can appreciate that pathogenicity is a non-binary phenomenon. It's not a matter of yes or no. It's a much more complicated set of interactions. So how might we categorize organisms that we encounter clinically? Either accidental pathogens, opportunistic or facultative pathogens, or obligate pathogens. It's really a spectrum. So is an organism pathogenic? Is a host susceptible to an infection? Is the environment permissive to the infection? These are all factors that go into determining what that host-microbe interaction is going to look like. The damage response framework is a really useful model that's been proposed by Arturo Casadevall. What Arturo proposes is that we see disease when we have an either inadequate immune response or too strong of an immune response. And so in this figure, we can see on the y-axis damage to host going up from the bottom, and immune response from 0 on the far left up to a very robust response on the right. The blue parabolic curve demonstrates this relationship. What level of damage do we see with each level of immune response? So too little immune response, and we're going to have an overwhelming infection and disease. Somewhere in the middle here, we have a state of health. And then too much response, and we'll have damaging inflammation or allergy. What would be some examples of organisms that might fit each of these categories? Two that I could think of were Rhodococcus equi and Aspergillus. Rhodococcus equi is a cause of pneumonia in foals. It occurs in otherwise healthy animals that are approximately 12 weeks old, and it's most likely due to a decline in maternal immunity. So a decrease in the immune response is what leads to infection. We also see Rhodococcus equi infections in people who are transplant recipients, people who have cancer, or AIDS. Aspergillus is a fungus that's ubiquitous in nature. It's present in decomposing matter, and we see it as a cause of pneumonia and air sacculitis in birds, but also allergic airway disease in horses. So too much of an immune response can also cause problems. The vast majority of the organisms that we're going to be discussing in this class and that you'll encounter clinically are probably best described as somewhere on the opportunistic spectrum. They're able to cause an infection when they gain access to a normally sterile site. So Staphylococcus species is a really commonly encountered opportunist, causes a wide variety of infections from mastitis in cattle, urinary tract infections, surgical site infections. Dermatophilus congolensis is a cause of rain scald. This occurs when we have wet conditions where the skin is macerated and traumatized. And then E. coli, frequently an opportunistic infection, can cause bacteria and sepsis in most species, as well as urinary tract infections in our companion animals, and of course in people as well. All of this discussion so far might seem very academic and not very clinically relevant or applied. But actually, by understanding pathogenesis and thinking about how organisms cause disease and why disease results, you can get better insight into what's going on in your patient. So instead of seeing an animal with an infection and thinking, I have to treat the infection, I want your first thought to be, why does this animal have an infection? What is the presence of this organism actually telling you? Infections are frequently secondary to a larger problem or underlying condition. And so determining what that primary cause is really critical in addressing the needs of your patient. Is a urinary tract infection in a dog secondary to diabetes, ectopic ureters, transitional cell carcinoma of the bladder? Do we have infections that are resulting from poor management? Do we have a failure to adequately clean milking equipment leading to outbreaks of mastitis? Do we have overcrowding of pigs and a failure to dock tails leading to tail biting and ascending infections of the spinal cords? What is the presence of this infection telling you about what's going on in these animals' environments? And how can we mitigate those effects? How can we correct the underlying issue? Another concept that I think oftentimes feels very academic, but is actually very directly applicable, is the epidemiological triad. And the whole concept of the epidemiological triad is that disease occurs when we have a favorable environment, a virulent agent, and a susceptible host. And while this might sound really obvious, if you drill down and think about each one of these, I think it can help you to sort of put together the pieces of a puzzle when encountering an infectious situation. So from the host perspective, do we have herd immunity? What is our individual susceptibility like? Do we have adequate nutrition, comorbidities, age? From the perspective of the agent, do we have the presence of an organism that possesses the requisite virulence factors? Is it there in sufficient numbers? Or have we perhaps had a change in virulence? And then environmental factors, from things that may seem really obvious, like are the host and agent actually in the same environment? Do we have favorable conditions, such as flooding versus drought? Do we have the presence of the required vectors or reservoirs of the infection? Or have we had some sort of change in the environment that facilitates the spread of the disease or alters host susceptibility? A recent example that I can think of that really exemplifies the epidemiological triad is the situation in Haiti since the earthquake in January of 2010. There was a 7.0 earthquake on January 12th, 2010. 200,000 people were killed and over 1 million people were displaced. The damage was really absolutely massive. You can see from these images here what was happening in the cities and how aid had to be dropped in because airports were also destroyed. 10 months following the earthquake, cases of cholera started to be identified, and the outbreak peaked with over 350,000 annual cases in 2011. So from the perspective of the epidemiological triad, we had a highly susceptible host. We had over 1 million displaced persons who were previously cholera negative. We had a naive population who hadn't had exposure to the pathogen, and so they lacked pre-existing immunity. We had a virulent agent, Vibrio cholera, which is a waterborne disease that had tragically been brought to Haiti by aid workers. And we had a very permissive environment, devastated infrastructure in an already quite desperately poor country, and a lack of water sanitary systems. So both sewage and drinking water were oftentimes untreated, which allowed this waterborne agent to spread without control. A lot of effort went into controlling cholera in Haiti, including a vaccination campaign. And Haiti was ultimately declared cholera-free in February 2022 after there had been no cases for 3 years. But later that year, 2 new cases of Vibrio cholera were identified, leading to a subsequent outbreak and over 20,000 cases as of early 2023. In this report from the Centers for Disease Control, the Morbidity and Mortality Weekly Report, they highlight a number of the social determinants of health that facilitated the development of this secondary outbreak, such as social unrest and poor public health infrastructure. So we've discussed host-microbe interactions and disease as an outcome, but what determines host spectrum? What makes an animal a suitable host for an organism? Why do some organisms tend to only infect certain species? We definitely see species associations. And why does an organism interact differently with different animals or different animals and people? I don't have a lot of specific answers to these questions, but I would encourage you to think about these in terms of shades of gray. While particular organisms may be associated with particular species, we can certainly see infections in other animals as well. So, for instance, Mannheimia hemolytica, common pathogen of the bovine respiratory tract, and in ruminants, we rarely see infections in people, but infections have been reported following bite wounds. So direct inoculation is a great way to have an overwhelming load of the pathogen at a site of infection. When considering host spectrum, we're often concerned with whether a particular organism has the capability of causing disease in people, whether it's a zoonoses. And a zoonosis is defined as an infectious disease caused by a pathogen that's jumped from a non-human animal into a human. Zoonotic diseases can be caused by any type of pathogen, so bacteria, fungi, viruses, prions, etc. And they're very, very common. It's been estimated that 6 out of every 10 infectious diseases in people had spread from animals. And to me, this really just exemplifies how common interspecies transmission really is. So what might be some examples of zoonoses? What diseases come to mind when you hear this term? Are there certain populations of people who are particularly at risk? And what might we see here in Western Canada? Maybe pause this video for a minute and think about these questions before continuing. Major themes that I see or have observed in discussions of zoonoses are things like SARS-CoV-2. Certainly, there's been a lot of discussion of that over the last few years. Foodborne organisms, so things like salmonella, really, really scary things like Ebola, tropical diseases, or the exotic pet trade. These tend to be the types of scenarios that come up in discussions of zoonoses. So is this problematic? Is there an issue with that? I think there really is. And I think the main issue is that this views disease from a very anthropocentric point of view. Humans aren't a goal. Organisms are simply looking for an ecological niche where their metabolic requirements can be fulfilled. And so I think it's much more useful to think like a biologist and think about diseases or organisms transmitting between species rather than from all of animals into people. And there's actually some really fascinating examples of diseases moving in the opposite direction. So Taenia solium, a parasitic disease, is one that we know a lot about, perhaps because parasites have very defined life cycles with definitive and intermediate hosts. In the case of Taenia solium, humans are the definitive host. So we have the adult tapeworm living in our guts. And when those eggs pass into the environment and are consumed by a pig, they develop muscle cysts. We then become reinfected when we eat those muscle cysts in uncooked meat, and the cycle continues. So we have transmission from both a person into a pig and a pig into a person. Both are necessary to complete the life cycle. We also see transmission between animal species. So canine distemper virus is a great example. It's threatening a number of endangered species, including the black-footed ferret in North America, where canine distemper has an approximately 100% mortality rate. Think about all the interesting biology we miss when we only really consider interspecies transmission in the direction of animals into people. There's a lot of fascinating interactions going on out there, not to mention all of the conservation and ecological implications. So I don't think we should privilege discussions of interspecies transmission to a human perspective, whether we're talking about zoonoses, reverse zoonoses, anthroponoses. There's a whole glossary of terms that are used. Like I said, transmission between different animals is important. I think as clinicians, it's important to think about transmission from the organism's perspective. And really just overall, I find the tendency to classify potential pathogens as zoonotic is just very reductive. So think like a biologist. So after today's lecture, just 2 questions. What are Koch's postulates and the 3 components of the epidemiological triad?